Y'all ready for some snow tonight? Yeah, did you see? Tomorrow should be snowy if everything cooperates. We went from sort of winter to summer, back to spring, and then now we're in winter again. Okay, a couple questions. Why do fathers take an extra pair of socks when they go golfing? In case they get a hole in one. Nice. What do a tick and the Eiffel Tower have in common? What do a tick and the Eiffel Tower have in common? They're both parasites. Oh, okay. What do you call a fish wearing a bow tie? Sophisticated. You like that? No? All right, tough crowd tonight. Tough crowd. Okay. Or what's happening Friday? What's significant about Friday? Chapter 12 quiz is due Friday by 11:59. So get it done before you leave for spring break, okay? Just so you don't forget. Get it done. If you're leaving Friday early, maybe do it tonight or tomorrow. You're going to be snowed in anyway, so, okay? So this is where we left off with muscle physiology. This is our last lecture in muscle physiology, and then we're going to take a little break. You do a quiz on Friday. Next week, as far as I'm concerned, you can take the whole week off. Okay, I don't know what's going on with the other classes, but and then when we come back, we'll be in the central nervous system, brain, and spinal cord. Okay? So what we got to finish up today are some of the details around muscle contraction. How many are feeling pretty comfortable with the way that actin and myosin interact? Let me see, by show of hands, pretty comfortable. Like if I had a pop quiz right now, you'd do pretty okay? Like, ooh, snap. Okay, that's fine, no pop quiz. It was just trying to stress you out. So I think actin and myosin and their interaction is a really important thing to review. How about the homework on the sarcomere? And the different segments, the different areas, the zones, the regions. How many are feeling pretty good about that? Okay, 3, not as many. 4, okay, great. So those are 2 really important topics. And you know, they're chock full of images and pictures that I could point to with arrows and say, what is this? What does it do? Like, for example, on an exam. Okay? Exams, what, a couple weeks away? It's on a Wednesday. It's not on a Monday. A few weeks away because next week is nothing. And then we have central nervous system, brain, and spinal cord. And then basically like the Wednesday after the Wednesday we get back from spring break is exam 3. So a little bit of time to digest this material, but this is your last week in this material. I think I'd take advantage of SI and TA sessions so that you can really hone in on the actin and myosin interaction. So in this slide, what we're showing is we're actually showing on the y-axis tension. Tension or force. Tension's a measure of a type of force. And then here on the x-axis is the sarcomere length in microns. And what I want you to be able to appreciate is the maximum amount of force or tension that is generated, right? The highest value is in this window of optimum resting length where you have maximum overlap of actin and myosin. So if you have the myosin heads, you can kind of appreciate the little blue or purple spheres or the little globular heads that are finding the actin filament. When you have the most amount of overlap between those 2 myofilaments, is when you can generate the maximum amount of force or tension. That should be pretty intuitive. Does that make sense? If you look at shorter sarcomere lengths where you've got some, the sarcomere of the Z to Z disc or the Z to Z line is the shortest. So this is an overcontracted state. So the analogy would be like right here at the biceps, you're generating lower amounts of force than you are mid-cycle. Right in the mid portion of the flex of the bicep is where you have the maximum overlap, and that's the window where the muscle is designed to give you the most force production or the most tension. So over here, you've got too short of a or too small of a sarcomere. You've got it overly contracted or overly crowded. You've got a lot of actin that's not seeing the myosin heads. And so you get less force production. Likewise, on this part of the curve, if the Z lines or the Z discs are further apart and the sarcomere is overly stretched, then you have very little overlap. And again, you have decreasing force production. So the take-home message is there's an optimum window or an optimum amount of force production when you have maximum overlap between actin and myosin. So the central nervous system, the CNS, that we're going to talk about when we get back from spring break, the CNS actually maintains some amount of basal tone in your muscles. Some amount of basal tone. And this muscle tone that's at a basal level is called tonus. Tonus. And that's because you want to have signals periodically going out to the muscles so that they stay ready and poised for contraction. So the computer analogy, many of you aren't computer experts, and neither am I, but you have an IT problem and they're trying to find your computer. They can do what's called pinging your computer, right? They can send a signal to your computer on the network, and your computer will actually send a signal back. It's kind of the same thing here, is the central nervous system kind of pings your muscles on a pretty regular basis to maintain some basal muscle tone so that it's you're ready to contract at all times. The relationship of this overlap is what we call a length-tension relationship. So the length of the sarcomere is directly related to the amount of tension or force that can be produced by the sarcomere. And right in the middle is that sweet spot. Now we have another relationship called this cross-sectional area relationship. And I don't have a picture for it, but the more cross-sectional area of a muscle means the more myofilaments, more actin and more myosin. Do you think that leads to greater force or smaller force? Larger cross-sectional area from larger muscles, greater force or smaller force? Greater force. Not a trick question. So that's referred to as the cross-sectional area force relationship. So the larger the muscle, the more force it produces. It's kind of a no-duh concept, but we like to label length-tension relationship and cross-sectional area force relationship. Okay, so now let's take a look at this next slide. We're going to break it up into a couple of pieces. We're going to talk about how muscles recruit muscle fibers. So there's a recruitment that takes place when you're performing an activity or exercising. We kind of use the analogy of when you get some feedback about picking up a pen versus picking up a water bottle versus picking up your backpack. You take a look at it and you're like, okay, this is going to be heavier than the pen that I just picked up. So immediately your central nervous system is going to recruit more myofilaments. And we've all been in that situation where you see a backpack and you go to pick it up and you think it's going to be heavier than it is, but it's like full of stuffing, right? And it's like super awkward. And you're like, oh wow, that was a lot lighter than I thought it was. Or the reverse. Someone throws you a bag and you catch it and you're like, what in the world is in this thing? Because you were bracing for it thinking it was going to be about the size of a backpack throw, and they have like, it's full of books and it's super, super heavy. So recruitment is variable depending upon the activity. Like, we're not going to be robotic and have the same contraction in every single activity. So first, what we need to define is a twitch. A twitch is the electrical impulse or a firing of a single action potential. That's a muscle twitch. So a single impulse, electrical impulse, or a single action potential is going to trigger one muscle twitch, one muscle firing to take place, or contraction event. And then the recruitment is the summation of multiple motor units. Remember, a motor unit is the motor nerve fiber and the muscle fibers that it innervates. And so you're going to recruit based upon motor unit needs. So, for example, if you have a stimulus to the nerve, and you can see this dotted line is the threshold, you've got a voltage stimulus, and it isn't until number 3 that it reaches the threshold. And then 4 through 9 are all firing electrical signals. And the corresponding muscle fiber, this is the nerve response, the corresponding muscle fiber, on the far left, this muscle fiber has myofilaments that are completely non-twitching or in a non-twitching state because you're below the threshold. You're below the threshold in 2, so there's no muscle fibers that are colored in red. In contrast, over at 7, 8, and 9, all of the muscle fibers are colored in red. They're all contracting. They're all twitching because it's this corresponding electrical signal that's the highest voltage stimulus recruiting all of the muscle fibers. So you go from 0 to all, 0 to all, and everything in between is some gradation of recruitment of different muscle fibers. Does that make sense? So I'm kind of showing you the left side and the right side because I know in the very back of the room, this figure might be difficult to see, but you have it on your printout or on your iPad. But what you're looking at is no muscle fibers are being recruited here on the left because the signal is below threshold. 7, 8, 9, all of these are at the highest voltage, recruiting all of the muscle fibers within that fascicle. Okay, now, if you look at the response of the muscle, this is the electrical signal of the nerve. These are the myofibers. This is the muscle itself. And we've got tension or force on the y-axis. And then we have our same 1 through 9. And you can sort of appreciate that 7, 8, and 9 are at maximum contraction because they've recruited all of the myofilaments within that fascicle. And the voltage signal going to the muscle is saying, give me maximum contraction. Okay? Pretty straightforward. So, a couple more concepts. This is a twitch. So here's a muscle firing, muscle twitches, muscle contraction, each one of these. Now, you can have them stair step. Before this one relaxes, you can actually fire the next signal to cause the muscle to contract, and we get this phenomenon known as trepe. And trepe is a staircase phenomenon that is additive. And why that is if you are, let's say, holding a tray and you're contracting your biceps brachii to hold a tray right here, and your friend's like, hold this for a minute, but let's say it's like 50 pounds, okay? 50, that's pretty heavy. And after about 2 minutes, you're like, okay, can I sit it down yet? Not yet. Okay, well, now I need to recruit more muscle fibers to hold that position. And so the trepe, or the staircase phenomenon, is your muscle's response to say, I need to sustain this force production for a longer period of time. And so that's how you staircase it up so that you can maintain that type of activity. I mean, you're not thinking about this. You're just trying to hold the tray for your friend, right? But that's what's going on between the central nervous system and at the local level of your muscle. Now we've got these two different phenomenons known as tetanus. And tetanus is from the ancient Greek, meaning tetnanos, meaning taut or tight. In some translations, it refers to tension, which is kind of like the same word, right? Some local dialects within ancient Greek. And so this is a phenomenon that allows for sustained muscle contraction, giving you the trepe that we saw in the upper right. Whereas if we look at incomplete tetanus, the muscle stays tight or taut or under tension, and you can nicely see this sort of trepe type of response as it continues. The reason it's incomplete tetanus is because it never stops. It keeps going. Complete tetanus is when it reaches a threshold and then you reach fatigue. And that's the big difference. So in the tray example of my friend, if I'm in incomplete tetanus, seeing trepe on the force production of the muscles, I'm holding the tray. The moment I'm like, I can't do this anymore, and it falls to the floor, that's complete tetanus because I've reached fatigue. Okay, so some other comparators to look at, isotonic versus isometric. So different types of contractions that we are able to produce. And remember, a contraction is a force over a length. That's what we do. So if you can appreciate this isometric at the bottom left, right? The muscle develops tension, but it does not shorten. Iso meaning same, metric meaning length. Same length. So you're holding the dumbbell still. Isometric. The muscle contracts, develops tension, but it doesn't change length. Iso meaning same, metric meaning length. Now, isotonic, we've got a concentric or an eccentric. Isotonic is the force is generated while either lengthening or shortening. So if you see isotonic concentric in the middle, we've got movement. Isotonic meaning same tension, isotonic. And you're getting concentric, meaning you're shortening the muscle, but the tension remains the same. So you're doing a regular curl, nice and smooth. And then on the lower right, isotonic eccentric. So this is like negatives, right, in the gym, where you're holding the dumbbell, and instead of just letting it drop, you slowly bring it down. So you maintain the same amount of tension, isotonic, and the muscle is lengthening, eccentric, while you're maintaining the tension. Okay, so isotonic versus isometric, and then isotonic has two different variations, concentric versus eccentric. Let's talk about how the muscles get their energy. So we're flashing back to the first unit. Remember, I've said numerous times that this class is cumulative. It's really important that you don't forget things that we've covered in the past. So our muscles, and where do we get our energy? Energy in the form of ATP. What does ATP stand for? Adenosine triphosphate. Nicely done. So on the far right, we see the breakdown in very broad brushstrokes. We see glycolysis at the top. We see anaerobic fermentation, yielding lactic acid, and in the middle. And then at the bottom, we see aerobic respiration in the presence of oxygen. That's why it's referred to as aerobic, because it needs oxygen to work. Someone remind us, if you go through glycolysis with no oxygen, approximately how many ATP net do you have? 2. Glycolysis yields approximately 2 net ATP, and you can appreciate it right here. You can circle it. If you run a sugar molecule, glucose, which is a monosaccharide, through glycolysis, and then in the presence of oxygen, you're able to enter aerobic respiration. How many additional ATP do we get net? 36. So now we have 36 plus 38, or 36 plus 2, which is 38. So aerobic is like upper 30s, right? Glycolysis is single digits too. So glycolysis, very little energy production, but it goes very quickly. Aerobic respiration, lots of energy in comparison to glycolysis, but it runs slower and it requires oxygen, which is a new demand. So what we're going to see is activities that you need high energy or quick energy are going to go through early forms of phosphate donations and glycolysis. Longer activities are going to go through aerobic respiration, right? So your heart likes to run on aerobic respiration because it runs all the time, right? The muscles in your quadriceps or in your hamstrings or in your biceps, they can run on glycolysis. There's some aerobic capability, right? But they can run on glycolysis. And so we're going to look at how this is broken down. So when muscles need immediate energy, we actually do what we call phosphate donations. Remember, moving the phosphate is where all the energy is, right? Adenosine triphosphate has 3 phosphates. Adenosine diphosphate has 2. Adenosine monophosphate has 1. So if you can see from this slide, in the muscle, we have a structure or a molecule known as myoglobin. And you can actually, in quick sprints, less than 10 seconds, you can strip phosphates from one molecule to another. So for example, if you want to make an ATP here and you're starting with an ADP, in the muscle, you can strip a phosphate off of myoglobin using myokinase and add it to your existing ADP and manufacture fast energy in the form of ATP. Likewise, in the muscle, you have an intermediate known as creatine phosphate. You can donate the phosphate and just leave creatine in the presence of creatine kinase, the enzyme, and this will add it to existing ADP to make ATP energy. And creatine phosphate is probably one of the more common, like muscle building energy drinks that a lot of bodybuilders use. It's for quick energy of less than 10 seconds, right? So 10 seconds would be like a deadlift or a bench press or a squat, right? And so you'll see a lot of those energy supplements in the gym utilizing these immediate phosphate donation energy demands. This is even before glycolysis. Okay, so now let's move to glycolysis, and eventually we'll get into aerobic. So immediate versus short term. So in the first 10 seconds of your activity, you're using the phosphagen system where you're donating phosphates. And then about the next 30 seconds, from like 10 up to 40, you're going to be utilizing glycolysis. And glycolysis doesn't just start here. It's going to start at 0. It just takes a little time for it to get going, right? And these things aren't like, one fuel tank is empty and the other one turns on, right? They kind of come on and they fade. They come on and then they fade. So there's this nice transition of energy. You don't see a sprinter run out of the gates and then and then they stop and then like, oh, now I hit glycolysis, right? So the body does it very, very smoothly where it's shifting to alternative fuel sources. So the immediate, the short term activities that are about 30 to 40 seconds of needed energy will utilize this glycolysis or this glycogen lactic acid system. Then the shift beyond 40 seconds starts turning into aerobic. After 40 seconds, we call this long-term energy. and oxygen delivery kind of catches up to the activity, and you utilize aerobic respiration. And then, of course, when the activity is done, there's still high respirations that are happening because you actually have this deficit of oxygen that you have to replenish. And you inevitably have a lot of buffering that needs to take place by the lactic acid that you created in the first 30 to 40 seconds of the activity. And one of the best ways to buffer acid in the body is to excrete hydrogen, free hydrogen ion, using the bicarbonate buffer system, which is it takes hydrogen ion and it binds it into a buffer system, a bicarbonate buffer system in the bloodstream. And what you produce is you exhale CO2 and water. And so when we blow out respiratory gas, it's CO2 that we exhale and water vapor. And you know it's water vapor, right? Because what do you do? How do you know it's water vapor? What's your evidence? How do you clean your sunglasses, right? And there's a little bit of water right there. So that is the repayment of that activity after the activity ends. Okay? So full circle. We introduced these concepts at the beginning of the semester in Unit 1. Now we're actually using them functionally to kind of explain how activities take place. So now we need to transition to talking about the other muscle types, smooth and cardiac. Because we've been spending most of our energy and most of our time talking about skeletal muscle. And so we're going to make a lot of parallels with cardiac because there's a lot of similarities. And then we're going to show where there's differences with smooth. And I just want you to be able to appreciate the two. So overall, going back to our tissue lecture, muscles function to contract or to shorten. They have a specialized architecture. They're cell-rich, they're highly vascularized, and the cells themselves are elongated or stretched out. They look like cylinders. They're not circular or they're not oblong or oval like what you probably drew in high school when you drew a cell, right? You drew like a round structure and then you colored in a black circle in the middle that was the nucleus. But they do have a lot of the same components that you studied in high school or in basic biology or in cell biology, right? They have these cytoskeletal proteins like actin and myosin. In other cells, they help keep the cell shape. They help for locomotion. They actually help for protein production and moving proteins that are made inside the cell to the cell surface. And inside of a muscle cell, they function for a lot of the specialization of contraction, actin and myosin, the myofilaments. We have our three types. We have skeletal, which we've studied extensively. We have smooth, and we have cardiac. Oops. So skeletal muscle, voluntary control, attaches to bones via tendons. This is a review slide. It's elongated, it's multinucleated, and it's striated. And this is the picture that we showed in the tissue lecture. And hopefully now you have a greater appreciation for the striations. The striations come from what? What gives the view of the striations? Why is it striated? Well, it's basically because of the sarcomeres, right? So it'd be the Z disc or the Z line to Z line, and the way the actin and the myosin overlap. You've got thick and thin areas, right, between your A band and your H zone and your I band. And so it gives you this very regular pattern of sarcomeres lined up. That's why it's striated. If we look at cardiac muscle, well, it's also striated. And you can see the striations. So do you think that it's organized in a similar way to skeletal with actin and myosin? It absolutely is. That's why it's striated as well. Under involuntary control versus skeletal, which is under voluntary control, it's branched, single nucleus, and it has these specialized intercalated discs. You can see here they're labeled with the arrow. They're these dark bands. Over here, they're kind of these dark bands. They're not labeled, but you can kind of appreciate them throughout this specimen. So it looks very much like skeletal muscle, behaves very much like skeletal muscle, and operates in a contractile event like skeletal muscle. Now, the anomaly is one more slide on cardiac. So no nerve stimulation was required, right? Because there's a pacemaker. So it's autorhythmic. It gives us the ability, because of the intercalated discs, for the propagation of the signal to happen from one cell to the next. We talked about that in our tissue lecture. I mentioned in our section on energy, it almost exclusively runs off of aerobic respiration because it's a long-term event. Your heart is contracting for more than 40 seconds, right? All the time. It's non-mitotic. Central dogma is a terminology that means the generally accepted viewpoint. The generally accepted viewpoint is that it doesn't divide. This is why patients have such problems when they have a myocardial infarction or a heart attack. So blood supply to the heart is cut off, right? Because a blood vessel is diseased and it's narrowed. So blood flow is restricted to the heart. The reason that that's important is because that's how the heart gets its oxygen supply. If it doesn't have oxygen, it can't run through aerobic respiration. So now the heart is starved of oxygen, can't generate the energy, and the cells die. And it creates what we call a myocardial infarction or a heart attack. Typically, right before that happens, the patient has chest pain. The word for that is angina, and that means chest pain. Because of the way the nerve plexus sits on the left side of the body, it's hard to pinpoint where the pain is, so they get shooting diffuse pain down the left arm with a heart attack. These are classic symptoms. Because of the central dogma of cardiac cells not regenerating well, after a patient survives a heart attack, I should say but if they survive and after they survive, what we worry about is the scar formation on the tissue. And if there's extensive scarring of the heart, then the heart's function is compromised. These might be terminologies that you've heard from talking with loved ones about, like, well, what happened? Well, Grandpa had a heart attack. What? He's okay. They're checking him out, but it looks like they're worried about a bunch of scar tissue. That's what they're talking about. Because when you tear a skeletal, how many of you have torn a skeletal muscle? In a room like this, yeah, okay. How long did it take for it to heal? Few weeks? Months? But it healed. Because skeletal muscle will divide and heal. Cardiac muscle will not. That's the big difference. Okay? All right, we're leaving skeletal and cardiac. Now I'm going to talk about a lot of differences, but I'll bring it back to where the similarities are that we've already learned about. So smooth muscle. Picture of smooth muscle here on the right. Uninucleated. We said it was fusiform. That means it's kind of thin at the tip, kind of wide in the middle, and then thin again at the other end. So that's the fusiform, kind of like this. Or a spindle. It's supposed to, another word it's used to describe it is spindle form. So this is found in the walls of the internal organs, body openings. It's under involuntary control, slow sustained contractions. No striations. So if I told you there's no striations in the tissue lecture, you just said, okay, fine, there's no striations. Skeletal and cardiac have striations, smooth muscle doesn't. Got it. Now I'm going to ask you a second tier question. Why do you think there aren't any striations? What's different? Give me some ideas. You don't use this type of muscle to move. I would agree. That's why it doesn't need to be striated. That's actually really good. What's causing it not to have the striated appearance? It doesn't have this, she said, does it not have Z discs? It doesn't have the same architecture. So the architecture is very different than in skeletal and cardiac. So let's talk about the architecture. It lacks Z discs, just like you said. Absolutely correct. It also lacks T tubules. It lacks the striations. So the sarcomere is organized completely different. But it still has some of the same concepts. Still has actin, still has myosin, and it still goes through contraction. But you can kind of appreciate in this picture of a relaxed, smooth muscle. So can you see that there's two muscle cells here? Thin, thick in the middle, and then it tapers down. Do you see that shape? That's the fusiform shape. And then here's a second one down here at the bottom. Over on the right are these two smooth muscle cells contracted. You can see that there's still actin, and we still have myosin. So there's still a thick and a thin filament, but they're organized more randomly or in like a framework or a network, not organized parallel lines. It does undergo mitosis like skeletal muscle. It can divide. And actually, that's one of the problems with a lot of disease states within blood vessels when you have atherosclerosis, is the smooth muscle inside the medial wall of the vessel is actually dividing, and you're getting lumen shrinkage over time. But the calcium, because calcium is so important, the calcium comes from the interstitial fluid, like outside the cell. It doesn't come from the terminal cisternae or the sarcoplasmic reticulum. And it still has actin and myosin. I just pointed them out to you. So you've got a relaxed smooth muscle, and then you have contracted on the far right. It's innervated in different ways. So it could be innervated as a multi-unit, which is shown on the left, A, multi-unit, or a single unit on the right, B. Multi-unit, these muscle fibers receive their own blood nerve supply. Larger arteries are multi-unit. Pulmonary airways, the iris in the eye, as well as the erector pili muscle in the skin, all multi-unit. And the nerve fibers, these autonomic nerve fibers, are coming down and innervating, and it still has these synapses like we talked about in skeletal muscle. Single unit, most of the rest of the body is single unit. Most of the rest of the smooth muscle in the body is like panel B, where you've got one nerve fiber innervating all of this muscle, and so all this muscle contracts as a single unit, whereas over here it contracts segmentally. So if we continue on this theme of smooth muscle cells and how they're a little different than what we've been learning about, number one is they can actually contract without a nerve stimulus, which is kind of crazy, right? So they're a little autorhythmic. And if you think about the GI tract, for example, the GI tract, you consume food, it moves through the alimentary canal, the GI tract, and it's going through slow, sustained waveform contractions as it moves the bolus of food throughout your intestinal system so that you can actually have time to absorb the nutrition. But it also has an autonomic nervous system innervation. That's autonomic nervous system, ANS. And we're going to talk about more of these types of terms when we come back from spring break, when we're into the central nervous system. But so here's a single unit smooth muscle, like we saw in the previous picture. You can see the autonomic nerve coming down, and it has these varicosities. And what do you think these varicosities are? What are they synonymous with? The varicosities. Look what's in them. You've got mitochondria, you have synaptic vesicles. What do you think the synaptic vesicles contain? neurotransmitter acetylcholine. So the varicosities are like the neuromuscular junction, right? And look at them all. All of these little bulges in this yellow autonomic nerve fiber are these varicosities. And of course, the diffuse junctions are like the synaptic clefts in skeletal muscle. So the gap between the varicosity and the smooth muscle is called a diffuse junction. But a lot of the same characteristics. So last, with calcium. So there's calcium gates on the plasma membrane of smooth muscle cells, and they're voltage regulated as well as chemically regulated or ligand regulated, as well as mechanically regulated. So calcium can enter into the smooth muscle via mechanical stimulation, voltage, or some ligand. Right? Think again about the GI tract. Right? So as the segment is actually contracting, it's causing the next segment to see the contraction and move. Calmodulin. Calmodulin is a structure that binds calcium, like calsequestrin, but it's called calmodulin within smooth muscles. And then the characteristics of smooth muscle that are very different than skeletal or cardiac is this slow contraction, slow relaxation, and a long latent period, or what we call a resting period in between. And again, you think about what it's being used for, right? It's controlling food throughout the, moving throughout the body. It's controlling, erector pili muscles, hair standing up. Controls the iris, the size of the iris, right? Or the opening through the iris. So slow period of relaxation, slow period of contraction, and then a long latent period where in between contractions, there's a lot of rest or non-activity taking place. Okay, let me ask this question to the group. In 97, this runner set the world record for the 800-meter dash in 1 minute, 42.67 seconds. As he was finishing the race, which energy source predominated in fueling his muscles? Feel free to talk it over with your neighbor. What do y'all think? Who's got an answer for us? Let's go back here. C, aerobically generate ATP. Why would you say that? He said it's because he's running for 1 minute and 40 plus seconds, so it had to be a longer time period of energy utilization because we're talking about the end of the race. That's exactly right. That's exactly right. So, I mean, an 800-meter dash is not a long run, right? But you're not using glycolysis the whole time. Okay, what is this picture an example of? What type of contraction? Who's got an answer for me? A, isotonic and concentric. That's right. Isotonic and concentric. Okay, how about this one? What is this gal doing? Holding plates of food, right? Which one is it? Who's got an answer for me? Somebody else? Over here. See? Isometric. Ideally, it's isometric, so your food doesn't end up on the floor, right? No movement of the muscle. Okay, how about this sequence of events? Focus on the triceps. Is it C? It's not C. There's movement taking place, right? What's that? B? Why is it B? The muscle's elongating? Yeah, you're maintaining force over an elongating or a lengthening muscle. Okay. All right, let's talk at the very end here in the last little segment about the types of muscle fibers. So I think this might fascinate some of you. How many of you are exercise science majors? Okay. So a little bit of exercise science pulling into this. And how many athletes, like even weekend athletes? That doesn't mean you're like sponsored or anything like that, okay? All right. So let's look at the way muscles are organized, okay? We have basic two main categories of muscle. Skeletal, we're back to skeletal muscle, by the way. So we've got slow muscles. I'm going to say slow muscles. Well, stay with me for a second. We have slow and then we have fast. So slow utilizes the oxidative or aerobic pathways to generate energy. That's why they're called slow. We refer to them as red muscle. They're called type 1 fibers. These are all names or aliases for slow muscle fibers. So also known as red muscle, slow oxidative type 1 fibers. You can see in this hematoxylin eosin prep, the slow oxidative are the darkly stained. They almost look black or dark brown, but they're red. They're really dark red. And the reason for it is because they have so many mitochondria and lots of capillaries and lots of oxygenation because they're utilizing aerobic pathways to generate energy. So they're slow, long twitch. They have less power than the other muscle fibers we're going to talk about because they have a different type of myosin. They have an isoform of myosin that actually moves slower in that video than what you saw. But these are for postural muscles, right? These are for respiratory muscles because they need to last for long, sustained periods of time. Do you understand why they need the mitochondria, the myoglobin, and the capillaries? Why is that important? Somebody link those together to our slow oxidative type 1 fiber. High mitochondria, more blood vessels, more blood flow, and more myoglobin. Any ideas? You want to take a stab at it? More energy production from aerobic pathways, right? So if you bring in more blood flow into the muscle, you bring in more oxygen. Hemoglobin's in the blood, myoglobin's in the muscle. Myoglobin strips the oxygen off of hemoglobin and brings it into the muscle. Mitochondria, because that's where, inside of the mitochondria is where aerobic respiration takes place. So you need more mitochondria if this muscle fiber is going to be relying exclusively on oxygen for aerobic respiration. But now look at this. Not every single one of these muscle fibers are slow oxidative or type 1 or red muscle, right? On this slide prep, it's like 40% are slow oxidative. 60% are fast glycolytic. And fast glycolytic is the second muscle type. So fast, short twitch, they're usually a lot larger than the slow. They're also known as aliases, are white muscle. Not really white, they're more pink. But, in comparison to the darkly stained, I guess anatomists initially thought these looked more white fibers, but they're really kind of pink when you stain them with eosin. But white muscle, fast glycolytic fiber, type 2 fiber. Okay. High power, but they're quickly fatigable. They leverage the creatine phosphate and the glycogen lactic acid pathways that we just learned about. But in this prep, maybe 60% are fast glycolytic in pink or in white, however you want to call that, and 40% are slow oxidative. So each muscle is going to have both types of fibers in them. So let's look at this. All right, this is showing slow oxidative versus fast glycolytic. And we're looking at marathon runners, swimmers, average males, sprinters, and jumpers. Ladies, I don't know why this textbook only had average males, but it did. Okay, sorry about that. You can send a message to McGraw-Hill. It's from their text. So let's look at the average Joe. Slow oxidative, 55, fast glycolytic, 45, fast glycolytic, 55. So almost 50-50. Now, if you train as a marathon runner, you favor the slow oxidative. If you train as a swimmer, you favor the slow oxidative. If you train as a sprinter and a jumper, you start favoring the fast glycolytic. Now, it doesn't go 100% in any of those circumstances, but 82% slow oxidative for marathon runners, that's a very high composition of slow oxidative fibers. So this is training specificity in your sport, right? That's why we do certain types of activities if you're going to be a sprinter, or certain types of activities if you're going to be a long distance runner. Does that make sense? All right. The picture does say it all, right? Muscle cells actually grow in size if you put pressure on them, if you stress them. They go through what we refer to as hypertrophy. So the red muscle adds more mitochondria and myoglobin. The red muscle is the slow oxidative. And they will enlarge. And smooth muscle does this, skeletal muscle does this, and so does cardiac muscle. And so we have these heart hypertrophies. If you put a load on the heart, you have a hypertensive individual, someone with high blood pressure. Over time, the heart is going to enlarge because it actually is a pump having to work against a greater force. So a heart hypertrophy is a clinical abnormality that usually gives you an indication that something's wrong. Okay. It also tends to be a side effect, probably for this guy, because I'm guessing he's using some type of anabolic steroid to enlarge the skeletal muscle. Well, do you think it has specificity only to skeletal muscle? It does not. And they'll have cardiac myopathies or hypertrophies, heart hypertrophies over time. And a great example of that, you can look it up online, is actually Arnold Schwarzenegger's had a couple of cardiac procedures to actually shrink the size of his heart. Well, they'll open up his chest, they cut out a portion of the left ventricle and then close it back up. It's almost like a tailoring procedure on like a dress shirt, if you will. Okay, so why do these muscles wear out? Why do they fatigue? Well, this is different than psychological fatigue, right? So for you athletes, your coach is like, 99% of your tiredness is in your head, right? Or whatever the number was. Like, get out there. So psychological fatigue is different. There is actually physiological fatigue. Now, there could be some examples here, all right? Calcium leakage into the sarcoplasm. An absence of glucose or glycogen. You've burned all your energy. On the energy theme, ATP is in scarce supply. If you release too much potassium, it lowers the excitability. Or maybe the neurotransmitter is exhausted. You haven't been able to recycle acetylcholine enough. Change in pH, like a lot of athletes think that the lactic acid burn is detrimental to their performance. There's a lot of literature that kind of demonstrates that you buffer it so fast. Once you, if you're contracting a muscle and you have tonic tone for a long period of time, you may be cutting off the blood supply. But the moment you relax and then recontract, you've just entered a whole new flow of blood. And that bicarbonate buffer system that we talked about will buffer that lactic acid in the form of free hydrogen ion at the level of the lungs on the next exhalation. So we talked about this briefly, but after the event, we have this period of time where we refer to it as oxygen debt, where we've got to move lactic acid into pyruvic acid. Most of that gets converted back to glucose in the liver. And then we have to replace our oxygen reserves. We've got to reestablish myoglobin and hemoglobin saturation with oxygen. And then we have to replenish any of our phosphates that got stripped off of different phosphate donations. So all of this is happening in the next 15 to 30 seconds after the activity, and your respirations stay elevated during that time. We refer to it as oxygen debt. When a muscle's at rest, which of the following is not associated chemically with the other? When it's at rest. Another way of saying that, which one is not bound to any of the others? Who's got an answer for us? Two of these? Which ones? Troponin and myosin? At rest, isn't troponin bound to tropomyosin, though? They're connected. So one of those answers is correct, though. Which answer is correct? At rest. Myosin's all by itself, right? At rest, in a relaxed state. Myosin is not bound to actin. You with me? There's no cross bridge formation. All right, can you label these? That was kind of your homework assignment. I'm going to kind of revisit that. Take a look at this. These are really valuable figures for you to be familiar with. Any questions before I have one last thing for you? Any questions? We good?